Madam Gladys, and I'm going to take you to today's lesson, which is biology techniques. And what did you learn last time? Who can remember what we learned last time? By show of hands, please. Yes?
Types of light microscope.
Types of light microscope. Someone else, can you clap for him? Someone else? Yes?
How to care for my
Very well. Anyone else? Yes? Yes? Be audible? Very well. However, today we have a class register. I want to confirm your attendance. If you hear your name, please raise your hand up. Dumie Isabella, Mutinda Jacqueline, Dennis Fondo, Muli Mukiti, Jane Evelyn Bulo, Makena Ann, Mwasia Joy, Havik Moireki, Sandra Joel, Bradley Mwangi, Zaid Juma, you can say present, Nyaguti Rodney, Geshunju Joy, Shanice Modoni, Mombi Alice Wamboi, Muigai Mary Njeri, Kerichi Mary Wanjiko, Moshira Patrick, Buru Sylvia, Monica Nyambura, Motuko Christopher, Nziva, Geshuge Evans, Kirika Manasseh Ngugi, David Janet, Masharia Tabitha, Kehia Stacy Mugoni, Kehia Stacy, Kanana Lizrita, Ndongo Sylvia, Kyoko Cesarin, Maureen Mukiri, Samuel Degwa, Mweha Penina Waroro. Is she present? Mboro Lucy Nyambura, Nelly Amondi, Nyamo Peace Wamboi, okay. Sakwa Barbara Nyondo, Stella Chelagat, Wanjiko Agnes, Lydia Damangala, Namoso Jael Akelo. Last time you make sure you confirm where they are and you tell me. However, we have a form today that you're going to fill in later in the class for the recording session. So our today's topic will be perform psychological techniques. Perform psychological techniques. And the subtopic will be definition of cytology, define cytology, and also cytological techniques. So our topic will be perform cytological techniques, and our subtopic will be to define cytology. Who understands better what psychology is? Who has an idea what is the meaning of psychology? Yes?
Study of cells.
Study of cells. Can you clap for her? Anyone else who has a better explanation? You can write this down. Cytology is a branch of biology. It is a branch of biology dealing with the structures. It is the branch of biology dealing with the structures, function, multiplication. It is a branch of biology dealing with the structure, function, multiplication, pathology, 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 and life history. Pathology and life history. It is a branch of biology dealing with the structure, function, multiplication, pathology, and life history. So basically, it is the study of the cell. So who can give an example of the cells that exist or that you know? Anyone who has an idea of the cells that are present, that exist in biology? Yes, Evans? Red blood cell. Can you clap for her? For him? Clap for him, clap. Any other? With the red blood cells, yes? Be audible? White blood cells. Can you clap for her, class? Any other? All these are from the animals. Any other example that exists? Yes? Platelets. Clap for him, class. Any other example? Yes, the one at the back. Mitochondria. Clap for him. So basically, as you have seen, it has been mentioned, you have the red blood cells, the white blood cells, the platelets, and the mitochondria. When you observe all these, you can find out that these are cells that are present in what? Anyone with an idea where they are present? Please raise your hand. They are present in the? Yes. Be audible? In animal cells. However, there are other cells that do exist that you have not mentioned, and it is the plant cells. Can someone give an idea of what is present in the plant cells? Any type of organelle, any type of structure present in the plant cells that you can remember even from your high school biology? Yes? You have said palisade cells. Can you clap for her? Clap. Any other? Any other type of cell, organelle present in the plant cells that you can remember? Yes? There is the cell wall. Any addition, any remembrance of any other organelle? Any other? In biology, the plants, what do they consist of? Any organ? We have the cytoplasm that is present also in the cell. So basically, when it comes to cytology, we can say, we can write it down. In cytology, the cells are divided into two major groups. In cytology, the cells are categorized into two major groups. Mainly plant cells. One of them is the plant cells. The second one is the animal cells. The animal cells. You can also write this down. The animal cells, they are larger in size. Sorry. The animal cells, they are larger in size than the plant cells. Another sentence. In order to carry out any research studies regarding the cells, we are required We are required to perform cytological techniques. We are required to perform cytological techniques. You can briefly define cytological techniques. The next sentence, cytological techniques. Cytological techniques. You can write down this. Cytological techniques refers to the method. It refers to the methods that are applied. It refers to methods that are applied. Methods that are applied in the study of cells. In the study of cells, their structure. The study of cells, their structure, function, study of the methods applied in the study of cells, their structure, function, and behavior. Structure, function, and their behavior. These techniques, another sentence, these techniques are essential. These techniques are essential for disease diagnosis. These techniques are essential for disease diagnosis. These techniques are essential for disease diagnosis, understanding cellular processes, understanding cellular, from the word cell, cellular processes, e.g., example of cellular processes, e.g., is mitosis, meiosis, right, etc. Example of cellular processes here include the mitosis process and the meiosis process. So you are in understanding cellular processes that have been mentioned, and carry out research in cell biology, Cell biology. So when we say they are cellular, cytological techniques are methods used to study cells. When we study cells, are cells organelles that are too big or too tiny? Anyone? Should they answer to raise their hands? When we say the cell, basically, it's the basic unit of life. So are they expected to be smaller in size or large? Anyone with the answer to raise their hand up? Yes? Small. So basically, you can clap for him. So cells are usually so tiny for the naked eye. I've mentioned this one before, right? What does it mean to observe cells? Not to be able to see the cells because they are too tiny for the naked eye. What does the term naked eye mean? Naked eye? Yes? Pardon? Speak up, please. Yes, with our own eyes, without any use of microscopes or eyeglasses. You recall this?
Yes.
In order to view the cell or to study cells, various instruments instruments or tools are usually used in the laboratory? Who can identify or recall the tool that is used in the study of cells or to view the cells in the laboratory? The tool that is used. Tool used. We had mentioned earlier in our previous class. Yes? Can you clap for her? So we use the microscope to study or to view the cells. In our last class, we did mention something about the microscopes. Someone mentioned about the light microscope. So when it comes to the light microscope, what is essential for one to utilize the light microscope? What do you use or what must be present in order to use the light microscope? Yes?
Light.
Be audible enough?
Light.
Can you clap for him? So light is one of the requirements. Light is one of the requirements needed in light microscopes or light microscopy. However, in order to perform these technological techniques, majority of the techniques usually have to use a certain type of a microscope, and one of them can be the light microscope, due to the fact that we require light in order to do what? To view. So you can write this down. So some of the tools used in cytological techniques, is light microscopy or light microscopes. Some of the tools used in cytological techniques is light microscopy. Light microscopy. Light microscopy. Under light microscopy, we have a number of microscopy or other microscopes that exist. So this is basically a broad term comprising of all the other microscopes that tend to utilize the light when viewing. So the first example under light microscopy or microscope is the bright field microscopy. Bright field microscopy. Bright field microscopy. Bright field. Bright field microscopy. As the term suggests, who can try to define what the term bright field microscopy entails? Ivan, can you give a try? Defining the bright field microscopy, what it means. A brief explanation. Yes? Can you clap for her? That's a good trial. You can write this down. In bright field microscopy, it entails the use of light. Into brackets, visible light. It entails the use of visible light. To illuminate. The cells stained with the dyes. To illuminate the cells stained with the dyes. Thus allowing observation of cell structures. And their organization. So far so good. Is that point clear? Anyone who has an issue? Any clarification? Okay, we continue. The second one, under light microscopy or light microscope, is the phase contrast. Phase, the word phase, with the PH, phase contrast, phase contrast. Phase contrast microscopy. Phase contrast microscopy. Phase contrast microscopy. Under this you can write, it enhances contrast, it enhances contrast in transparent specimen. It enhances contrast in transparent specimens without staining. It enhances contrast in transparent specimen without staining. Without staining. After staining, you can do a semicolon, and you write staining. Staining refers to the technique or method. Staining refers to the technique or method of using dyes, or a colored substance, of using dyes or colored substance to apply to the cells, to enhance, better viewing, to apply to the cells to enhance better viewing. Then the next sentence under phase contrast, you can say it is used in viewing live cells. It is used in viewing live cells. It is used in viewing live cells. Full stop. The other example is the fluorescent microscopy. Or rather, the other category under light microscopy is the fluorescent microscopy. Fluorescent microscopy. Okay, right, it uses the UV light. It uses the UV light. It uses the UV light. It uses the UV light as its light source. Uses the UV light as its light source. Uses the UV light as its light source. Full stop. When the UV light hits on an object, when the UV light hits on an object, it excites electrons. It excites electrons of the object. It excites the electrons of the object. Then full stop. The other category of the light microscope is the confocal microscopy. Confocal microscopy. Can you hear me at the back?
Yes.
Okay, confocal microscopy, you can write this down. It uses laser to focus. It uses laser to focus on specific planes. planes. Uses laser to focus. It uses laser to focus on specific planes of the sample. It uses laser to focus on the specific planes of the sample. Eliminating. From the word eliminate. Eliminating any light that is out of focus. Eliminating any light that is out of focus. Eliminating any light that is out of focus. This leads to a sharper image. This leads to a sharper image, especially in thick specimen. You can write after specimen, you write into bracket item view or object viewed. Into bracket object viewed. That's the meaning of the specimen. And repeat the sentence. It provides sharper images, especially in big specimens, and often used with fluorescent staining. From the word fluorescent, fluorescent staining. The last or the other example is the electron. Transmission electron microscopy. From the word transmission electron microscopy. You can write this down. Electrons pass through these sections. Electrons pass through thin sections. Electrons pass through thin sections of cells, creating a clear detailed image, providing a clear detailed image of organelles. Providing a clear detailed image of organelles. Organelles in either plant cells, of organelles either in plant cells, of organelles either in plant cells or the animal cells, e.g. we have mitochondria, e.g. mitochondria and nucleus. E.g. mitochondria and nucleus. The other one you can write is the scanning electron microscope. Scanning, from the word scanning electron microscope. Scanning electron microscope. Scanning electron microscope, also abbreviated as SEM. The first one was abbreviated as TEM into brackets. TEM. The first one was TEM. The second one is SEM. You can write this down. Electrons are scattered. Electrons are scattered. Electrons are scattered off the surface. Electrons are scattered off the surface of samples. Electrons are scattered off the surface of samples, providing detailed providing detailed images. A brief assignment. Can write this down. Assignment. Discuss the application. Discuss the application of cytological techniques. Discuss the applications of cytological techniques in microscopy. Discuss the applications of cytological. The applications of cytological. Discuss the applications of cytological techniques in microscopy. Discuss applications of cytological techniques in microscopy. 10 marks. And this one will be due next lesson, next week at the same time. I think it will be on Tuesday. You hand it over your assignment by Tuesday next week from 8 a.m. Any questions so far?